This is the first lecture for Chapter 17 on adaptive immunity. I have already gone over the innate immune response in Chapter 16, which, if you remember, is composed of two lines of defense. The first line of defense is designed to prevent infection, to keep the microbes out of your body, and this involves things like physical factors, chemical factors, washing processes, and your normal microbiota. Your innate immune response also involves your second line of defense. So this occurs once the microbes have gotten past your first line and they have entered into your tissues. And this involves reactions from phagocytic white blood cells, inflammation, fever, interferon, and complement. Now, Chapter 17 is about the other part of your immune response, which is the adaptive immune response. Your third line of defense is referred to your adaptive or specific immune response, and these two words are important. This is the immune response that is adapting to an infection that you have. And it is also a response to that specific microbe, that specific strain of the microbe that is infecting you. So your third line of defense is your response to a specific microbe that has infected you. And this involves your T cells and B cells and also the antibodies. The other important thing about your third line of defense is that this immune response has memory. So your white blood cells are able to remember the microbe that has infected you and protect you from future infections. Your adaptive or specific immune response is composed of two parts. The first part is called the humoral immune response, and we refer to it as humoral because it's referring to a humor you find in the body, and humor is an old-fashioned word for liquid, and the humor it's referring to is the plasma. And the immune component that you find in the plasma that relates to your third line of defense are the antibodies. So antibodies are found in the plasma, the liquid part of the blood, and they are produced by B cells. So B cells are also associated with the humoral immune response. Your humoral immune response is very good at protecting you from bacteria, viruses, and toxins. The second part of your adaptive or specific immune response is the cell-mediated immune response. Cell-mediated, this is referring to T cells. So T cells are the main mediator of your cell-mediated immune response. And what the T cells basically do is they kill eukaryotic cells. So your cell-mediated immune response is very good at protecting you from parasites and from intracellular microbes that are found inside your cells. So the T cells are very effective at killing virally infected cells and other bacterially infected cells. Also, your cell-mediated immune response is responsible for eliminating cancer cells and also for transplant rejection. For this lecture, I'm going to focus on the humoral immune response and more specifically just on antibodies. In order to understand antibodies, there are three important terms you need to know. The first term is antigen. An antigen is a molecule that stimulates an immune response. So an antigen is a molecule that is going to activate or trigger an immune response. And most antigens are some sort of large protein. So they have to be relatively large molecules and they have to have a protein component. So that's what an antigen is. And antigens can be pretty much any foreign protein. So a lot of the proteins you find on the outside of microbes can act as antigens, can stimulate your immune response. Also foreign proteins such as pollen or dander, can also act as antigens. The second term you need to know is epitope. An epitope is defined as a small part of an antigen where an antibody binds. So an epitope is a smaller part of an antigen, and that is where the antibody will bind. Every antigen has many epitopes. The third term you need to know is antibody. And the definition of an antibody is a serum protein. So a protein that is found in the serum of humans, which recognizes an epitope. So when we say recognize, we mean it binds to an epitope. So these are three important terms you need to know: antigen, epitope, and antibody. And you need to know how they relate to each other. So an antigen stimulates an immune response, and it's some sort of protein. Every antigen has several epitopes. So the epitope is just a small part of the antigen. And the antibody is a serum protein that binds to the epitope of an antigen. Here is a diagram which relates those three different terms to each other. And here you have antigens. And in this example, the antigens are found on the cell wall of a bacterial cell. So this large molecule is one antigen, and there are several different antigens on this bacterium. Every antigen has several epitopes, actually 3 epitopes. So this would be one epitope, this is a second epitope, this is a third epitope. So every antigen on this bacterial cell has 3 epitopes in this example. The antibody recognizes one epitope on the antigen of the microbe. And antibodies are proteins, so if you remember learning about proteins, proteins are highly specific. So every antibody recognizes only one epitope. So this antibody A only recognizes and binds to that triangular epitope, whereas antibody B in this case only recognizes and binds to that rounded epitope. So every antigen has several epitopes, and the antibody binds to one epitope on a particular antigen. The next thing I want to go through is basic antibody structure. And in most diagrams, antibodies are drawn as green Y structures, but there's more detail in this that you need to know. This is a more detailed diagram of a basic antibody structure. And in reality, every antibody is composed of 4 protein chains. So 2 heavy chains and 2 light chains. The heavy chains are called heavy chains because they are larger, and the light chains are called light chains because they are smaller. The 2 heavy chains are identical to each other, so they have exactly the same structure. And that's the same with the light chains. The 2 light chains are identical to each other. And the 4 chains are held together by disulfide bonds, which help to stabilize the structure of the antibody. Each chain, heavy chain and light chain, has 2 regions. It has a constant region and a variable region. The constant region of each chain has the same structure in all members of a species. So if you look at all the antibodies that are in my body and compare the constant region to the antibodies that are in your body, they would have exactly the same structure. So the constant region of the light chain and the constant region of the heavy chain have exactly the same structure in all members of a species. The variable region is called variable because the structure varies. You get different types of structure in different antibodies. So the variable region will have different structure in the different antibodies in one individual. Maybe the most important part of the antibody are the antigen binding sites. So the antigen binding site is where the antibody will bind to the antigen. And that site is a combination of the variable region of the light chain and variable region of the heavy chain. So here it is in bigger detail. Here you have the variable region of the heavy chain, variable region of the light chain, and together they create the antigen binding site. So this antigen binding site is going to bind to the epitope of the antigen. So kind of like an active site for an enzyme that has a particular shape, the antigen binding site for the antibody will have a particular shape, which will fit perfectly an epitope of a particular antigen. So antigen binding sites, this is where the antibody can bind to the antigen. And because the heavy chain and the light chain have exactly the same structure, the two heavy chains and the two light chains have exactly the same structure, one antibody can bind two particular epitopes. And one last thing about antibody structure is on the heavy chain, constant region of the heavy chain, there is a complement binding site. So that's not written on this diagram, but on the heavy chain constant region of the antibody, you have a complement binding site. Another way to look at an antibody, here is an antibody. So this is the antibody, the basic Y structure. Here is the antigen binding site. And for this particular antibody, it recognizes this epitope on that green antigen. And each side of the antibody recognizes exactly the same epitope. And antibodies are highly specific, so that a particular antibody can only recognize a particular epitope. So this antibody, again, recognizes that epitope, so if it comes in contact with a different epitope, it will be unable to bind. So this makes antibodies highly specific. Again, kind of like enzymes, the antibodies, because of their structure, they can recognize only one epitope of an antigen. So they're highly, highly specific. When we talk about antibodies, there are actually 5 different classes of antibodies. And when I refer to a class of antibody, it's based on the structure and the location and the function of those antibodies. And another term for antibody is immunoglobulin. Immuno refers to the immune system. Globulin is a protein. So immunoglobulins, and they're abbreviated IG. So the different classes of antibodies start with IG, and then they use letters which represent basically the sizes of the antibody. The first antibody, class of antibody that I want to talk about is IgG. IgG is immunoglobulin gamma. And this is the most common antibody. Usually when you talk about antibodies, this is sort of the classical antibody that you refer to. And IgG is found mainly in the blood. It makes up about 80% of the serum antibodies in your blood. And IgG is just a monomer, one Y structure. So it's relatively small, so it is actually able to cross into tissues. And IgG is very effective at killing or attacking bacteria and viruses and toxins. And something that will come up a little bit later that I want you to know is that it is the second antibody made by an activated B cell. And that will make sense a little bit later. The second class of antibody I want to go over is IgA. So IgA, this is immunoglobulin alpha. And IgA is actually a dimer. So it's two of those Y structures bound together at the constant region of the heavy chain. So IgA actually has 4 identical antigen binding sites. And IgA is found mainly in excretions. It's found in mucus, it's found in tears, and it's found in breast milk. And the main purpose of IgA is to prevent attachment. So remember when the bacteria, some bacteria have IgA protease that can break down IgA. That's because IgA prevents attachment of microbes, and specifically in the mucous membranes. The third class of antibody that I want to talk about is IgM. M stands for mu, but you can kind of imagine that M stands for massive, because this is a class of antibody that is composed of 5 monomers. So you have 5 monomers that are joined together at the constant region of the heavy chain. So since it has 5 monomers, that means it has 10 identical antigen binding sites. So all of these recognize exactly the same antigen. And IgM is only found in the blood. So it makes up about 10% of the antibodies that you have in the blood. And it is so large that it cannot leave the blood. So it is only found in the blood. And IgM is responsible for transfusion reactions. So if you give somebody the wrong blood type, the reaction that occurs is due to the IgM. And the last important thing about IgM is that it is the first class of antibody produced by an activated B cell. And again, that'll be important in the next lecture. The next class of antibody I want to talk about is IgD delta. And this is a little bit different of an antibody. It's a monomer, but this is not floating free. So actually IgD is attached to the surface of a B cell. So IgD actually acts as the B cell receptor. So this is attached to the surface of the B cell and it's acting as a receptor. And when the IgD binds to its particular antigen that it is specific for, it serves to activate that B cell. So that is the purpose of IgD. IgE epsilon, immunoglobulin epsilon, is actually very similar to IgD. It is not found floating free in the serum or in tissues. Instead, this is bound to the surface of basophils or mast cells. So mast cells are very similar to basophils. So the IgE is acting as a receptor on the mast cells or the basophils. And what will happen is when the IgE recognizes the antigen it's specific for, that is going to send a signal to the mast cell or the basophil to activate. And mast cells and basophils, when they are activated, they release histamine, which triggers inflammation. So these are the 5 classes of antibody: IgG, IgA, IgM, IgD, and IgE. So you should know their basic structures, where they are found, other characteristics, and what they do. As a review, this is a table that compares the 5 different classes of antibodies. So you have the names up here: IgM, IgG, IgA, IgE, and IgD. It tells you their number of binding sites, also the percentage in the serum, and of course IgG is the most common in the serum. And it tells you if it can cross into tissues like the placenta, and it also gives you their basic function. So far I have talked about the structure of antibodies and the different classes of antibodies, but the most important thing about them is how they protect you from infection. And when an antibody binds to an antigen, there are 5 different results of this interaction that are going to protect you from infection. The first benefit that occurs when antibodies bind to antigen is agglutination. So agglutination is the first result or the first protective mechanism that occurs when antibodies bind to antigen. And agglutination basically means clumping. So every monomer, so IgG for instance, is able to bind to 2 microbes that have the same epitope. And because every antibody can bind to 2 different microbes, you can end up with clumping together, getting clumps of antibody binding to microbes. And IgM is the very best at agglutination because it has 10 different antibody binding sites. So one IgM antibody can bind to 10 different microbes. The second benefit of antibody binding to antigen is activation of complement. So remember on the structure of the antibody, I added a complement binding site to the constant region of the heavy chains so that when the antibodies bind to the microbe, that will activate complement. And that is actually the classical pathway of complement activation. And when complement is activated, that leads to the acronym OIL. So remember, when complement is activated, you will produce opsonization. Opsonization is when the microbe is covered with the complement protein, and that leads to phagocytosis. Another result is inflammation. Some of the complement proteins will trigger histamine release, which will trigger inflammation. And the last benefit, L, is lysis, and that is performed by MAC formation, which the complement proteins form pores in the membrane of the microbe, and that leads to lysis. So activation of complement is actually going to lead to 3 results: opsonization, inflammation, and lysis. A third benefit of antibody activation is actually opsonization. So when I first gave you the definition of opsonization, I said it is when a microbe is coated by a protein. So that could either be complement proteins or it could be antibodies themselves. So if antibodies bind all over a microbe, then that is going to make it easier for the white blood cell to adhere to the microbe and phagocytose the microbe. Fourth benefit or result of antibody binding to antigen is neutralization. So neutralization means that if the antibody binds all over the microbe, it's going to prevent infection. So for example, if the antibody binds to all the protein fibers on this naked virus, then it is no longer able to adhere, recognize, and attach to a host cell. And if it can't recognize and attach to a host cell, it cannot infect the cell. So having the antibody bind all over the protein fibers or the spikes, if it's an envelope virus, will neutralize the virus, prevent infection. Also, IgA is really good at preventing attachment. So this shows IgG, but IgA is also good at preventing attachment. And if it binds all over the microbe, all over the capsule, all over the fimbriae, then the bacteria is unable to attach to the tissues and it will be washed out or flushed out. Also with toxins, exotoxins, because they are proteins, if the antibody binds all over the toxin, then it will be unable to bind to the target cell and have the negative effect on the target cell. So neutralization is a really important protective mechanism that antibodies have. And remember, your humoral immune response is very effective against viruses, bacteria, and toxins. The last benefit of antibody binding to antigen has a really long name, and it's called antibody-dependent cell-mediated cytotoxicity. And it's a really long term, but it's a very simple idea. So this is antibody dependent, so you have to have an antibody bound to the microbe, and it's going to stimulate cell-mediated cytotoxicity. So it's going to stimulate other white blood cells to kill the target cell. So this is basically effective against large microbes, eukaryotic microbes like parasites. So what the antibody does is it recognizes an antigen on the parasite and it binds to that parasite, and then that is a signal for other cells such as eosinophils, which remember are part of your second line of defense and they are anti-parasitic. It'll activate the eosinophils to attack the parasite. It can also signal to other white blood cells that this is an organism that has to be eliminated. So, antibody-dependent cell-mediated cytotoxicity. This is where the antibody binds to a microbe, and that is a signal that other white blood cells need to attack and eliminate that microbe. So, the 5 benefits of having antibody bind to antigen: agglutination, activation of complement, opsonization, neutralization, and antibody-dependent cell-mediated cytotoxicity. And notice that all the antibody is actually doing is just binding to the antigen. Antibodies are not enzymes. They do not catalyze a reaction. All they do is bind to the microbe. They bind to the target. That's all they do, and then that will cause different types of results.